Okay, so we are talking about the G0 phase. We're talking about the G0 phase or the quiescent phase.
Sir,
Hello?
I'm asking if you can repeat on G2.
We were still on G2 before your network cut off.
So under G2, we said the G2 is a second growth phase. Remember, the first growth phase is G1. So G2 is a second growth phase. And during G2, what happens that is reorganization of the organelles. There is reorganization of the organelles. So organelles are able to replicate. In addition to that, during the G2 phase, also the chromosomes are able to coil. So the chromosomes are going to coil and then they're going to shorten. This is in readiness for the cell to enter the mitotic phase. So that's G2. Second growth phase, reorganization of organelles via replication. They're going to replicate, coiling and shortening of the chromosomes in readiness for the mitotic phase or mitotic division, the M phase. And then after that, we have the G0 phase. G0 phase or resting.
Could you just go back to the slide we took?
Sorry?
Could you go back to the slide we take screenshot?
The notes will be shared.
All right, thank you.
So we have the G0 phase or the resting phase or the quiescent phase. So during this phase, the cell exists in a quiescent state. The cell exists in a quiescent state. So some cells in eukaryotes actually enter G0 indefinitely, such as the neurons. Or some enter G0 phase for simply a short period of time, such as the liver and the kidney cells. So we have other cells that actually never enter the G0 phase. So for example, epithelial cells, they never enter the G0 phase. So that's a G0 phase. And during the G0 phase, in short, the cell is actually neither dividing nor preparing to divide. So it leaves the cell cycle. So let's look at mitosis or mitotic phase. So this is a phase during which the cell will be undergoing division. So it's called mitotic phase or the M phase in short. And this results from the production of two daughter cells, or rather it results in the production of two daughter cells from a single parental cell. And these daughter cells are actually identical to one another and also identical to the original parent which produced them. So basically during the mitotic phase, two major processes or events take place. We have what is known as karyokinesis, which is simply nuclear division. And we have what is known as cytokinesis, which is division, cytoplasm, organelles, and cell membrane division. So we have chirokinesis, which is nuclear division, and cytokinesis, which is cell division. So these two major events happen during the process of the mitotic phase. And then when we look at the mitotic phase, it's divided in two stages. So we have the prophase, we have the metaphase, we have the anaphase, and we have the telophase stage. We have the prophase, the metaphase, the anaphase, and the telophase stages of mitosis. So usually the time that the cell spends in each of these phases are quite different, but prophase is usually the longest. Prophase is usually the longest, while metaphase is usually the shortest of these stages. So prophase is the longest, or is longer than the others, while metaphase is shorter than the rest. So metaphase is the shortest. So let's look at what happens during the process of prophase, during the prophase stage. As mentioned earlier, in accordance, this is the first stage of mitosis. During this phase, the nuclear envelope is going to start to dissolve. The nuclear envelope dissolves. The nuclear membrane is going to disappear. So there's a disappearance of the nuclear membrane. And then we see replicated chromosomes condensing and also becoming visible. So chromatin, during this stage, chromatin is going to condense into chromosomes by coiling and thickening. So the chromatin will condense into chromosomes by coiling and thickening. Hence, we see that the replicated chromosomes will condense and become visible. And then we see that the centrioles are going to separate and migrate to the pores of the cells. They're going to separate and migrate to the pores of the cells. So we are saying spindle fibers are going to appear from the replicated centrioles. They're going to appear. And then centrioles migrate to the opposite of the to the pores of the cells. And then the nucleolus is going to disappear as well. The microtubules are going to appear between the two centrosomes. They're going to appear between the two centrosomes also during the process of prophase. And then these microtubules of the developing mitotic spindle are going to attach to the chromosomes, to the chromosomes. So during this prophase period, the other thing is that there's no synapsis of the homologous chromosomes. There's no synapsis of the homologous chromosomes. And then after the prophase stage, we have the metaphase
Please
stage.
explain on
And
the homologous. Whatever.
So during the metaphase stage, so we looked at the homologous chromosomes during our last class. We looked at homologous chromosomes, and we said that a homologous chromosome is a pair of chromosomes which contain the same gene sequences, each being derived from one parent. That's a homologous chromosome. It's a pair of chromosomes that contain the same gene sequences, each derived from one parent. So for more of this, you can review our previous lesson where we looked at the chromosomal theory of inheritance. We looked at the structure of the chromosome, the discovery of the chromosome, and we explained each and every structure of the chromosome and their functions as well. So we are talking about the metaphase. And during the metaphase, the cell prepares chromosomes for division. So during metaphase, the chromosomes are going to align themselves in an orderly manner. They're going to align themselves at the center of the cell, otherwise known as the equatorial region. They are at a metaphase plate. They're going to align at the metaphase plate. there. That's what happens during metaphase period. And then the spindle fibers. The spindle fibers which are supporting the chromatids are also in position. So the spindle fibers attach from daughter cells to chromosomes at the central mere, And then we have the anaphase period. We have the anaphase period. During the anaphase, we have the chromatids. The sister chromatids are going to separate from each other. So the chromosomes or the chromatids are going to separate into individual chromosomes. And this will be assisted by the spindle fibers, which are going to pull the chromosomes apart. So the sister chromatids will separate from each other via assistance of the spindle fibers, which pull the chromosomes apart. So these are pulled at their kinetocores towards opposite spindle pores. So half of the chromosome, which are called the chromatid, moves to each daughter cell during the process of anaphase. Okay.